So today, we're going to talk about geomicrobiology across critical zones. We have two speakers, Dr. M Aaronson and Dr. Schaeffer. Dr. M Aaronson is an associate professor in the Department of Plant Pathology and Microbiology at the University of California, Riverside. Her research explores how dispersal and environmental filtering influence the formation of soil microbial communities. And she's the principal investigator of the Geomicrobiology Critical Zone Network Cluster. And Dr. Schaeffer is an associate professor in the Department of Biosystems Engineering and Soil Sciences at the University of Tennessee, Knoxville. His research interests including studying ecosystem and landscape scale processes and their link to environmental controls of physiology at the organism scale. He's an investigator with the CINet Critical Zone Network Cluster. So thank you both for joining the meeting and helping and presenting. And without further ado, let's get started. It's more like a presentation and discussion than an actual monologue. It's more of a dialogue. Probably, Emma, you want to get started?
Actually, I think we're gonna start this morning with Sean. It's a little early on the West Coast still, so.
Yeah, so along those lines, thank you, Christos, for that introduction. Are you asking folks to just chime in when they have a question, or should I just plow through and wait till the end for questions?
Actually, I think anyone can, in the meantime, as someone is presenting, ask some questions so that we can enhance dialogue. So you can start sharing your screen and presenting, and we go from there.
All right, here we go. I hope everybody can see that. I'm gonna go to the beginning. So yeah, thank you, Christos, and really enjoying this junior scientist organization. I think it's great that you've set up this resource. So I want to talk a little bit about my part in this critical interface network, and specifically some of the microbial dynamics across biogeochemical interfaces in the critical zone. And this may be familiar to a lot of you who are part of the CINet program, but I thought it was worth, at least for me, going over it again. So I took some of this language straight from the proposal. Thank you, Praveen and others, for summarizing it so well. But some of the key arguments and findings that led up to this present project were the recognition from the previous work at the IML-CZO that the dynamics of the critical zone aren't uniform across the landscape or over time, and that there seems to be processes operating at critical interfaces or biogeochemical interfaces can control or regulate material storage, transport, and transformations. Now that may seem a fairly obvious statement in hindsight, but there's an increasing realization that we need to identify these spatial and temporal scales to define these interfaces. So the previous work at IML-CZO identified sort of three major critical interfaces, those being the near land surface, so the soil surface to atmosphere interface, the active root zone, and also the river corridor. So this is where we have, I think, in terms of carbon cycling. So I think of it in terms of where carbon enters the ecosystem at this near land surface interface with plant growth. And then you have a strong plant soil interaction that goes all the way through the active rooting zone that controls the further vertical movement and storage of carbon and nutrients. But then there's also this strong lateral transfer of materials, usually downslope. So that means going from uplands to lowlands to possibly the floodplain and river corridors, which represents a major vector of export from ecosystem. So all that previous work at IMLCZO has driven some new hypothesis development for this current project. Again, I just took part of the overall hypothesis from the proposal, and that is that dynamics of critical interfaces exert a disproportionately large control on the overall dynamics of critical zones at the landscape scale. And the reason that we all think this is important is because this may help us to distinguish whether the variation we see in a system is a result of some dynamic equilibrium process, or whether there is actually a disequilibrium process occurring. So this is important, not just for me who works at a mechanistic and empirical scale, but for people who are designing models of ecosystem function and trying to gain some predictive ability. It's important to be able to understand the variation in a system, and if it is in some kind of equilibrium or whether it is being forced into a new equilibrium or is just overall in complete disequilibrium. That's very important in today's research. So I'm relatively new to this critical zone network. I wasn't necessarily involved in the previous work at the IMLCZO, so I appreciate all the feedback and collaboration that I've gotten so far. But my contribution as a newer investigator has been to take sort of a microbial-centric view of these fluxes of energy and nutrients across the landscape. So the way I see that is looking at microbial dynamics across spatial and temporal scales. And in this case, it means looking at both the lateral and vertical controls on microbial carbon cycling and possibly nitrogen cycling as well. And looking at those across some important spatial and temporal biogeochemical gradients. And the ones that I think we've identified for the CINet project are one, I put the examples here, this gradient across an erosional and depositional centers across the landscape. And that involves movement of material from summits and the tops of slopes down to possibly the river corridor. But there's also in these landscapes, there's lots of upland depressions where erosional materials can accumulate, and they have not only they have very different hydrological regimes that I'm finding out, getting to work with so many hydrologists, but they have very different carbon and nutrient contents, very different soil profiles, and all of those things can lead to different microbial dynamics that might control the stabilization or loss of that carbon over time. I'm also interested in the vertical movement of this material and how microbes are facilitating that across what we're calling the management induced reactive zone, or MERS. So this includes not just the influence of growing plants and rhizo deposits on soil processes, but also this effect of tile drainage, which is very important in agricultural systems, especially in Illinois, I'm finding out. And then I also want to look at these, especially the temporal dynamics related to climate and land use change. So for me, that means on the near surface in these erosional and depositional centers, and in the MIRZ, looking at how precipitation frequency and intensity are changing the balance, potential balance of carbon storage or carbon loss by microbial decomposition, but also some important human activities like tillage, a cropping system. We may be looking at comparing, say, a restored prairie to a traditional corn soybean rotation, and also this impact of tile drainage that I mentioned that very much changes the soil moisture profile and is therefore going to change microbial activity. So I put this conceptual figure of how I'm thinking about how carbon is perhaps moving across this landscape, both laterally and vertically. And how do we sort of encapsulate those microbial dynamics? We can do that by sort of sequestering or partitioning soil carbon into different forms, whether it's complex, either chemically complex or somehow protected in the form of mineral associations or locked up in soil macroaggregates, versus more simple forms of carbon, which are the ones that are more readily utilizable by microbes. And so how they're taking direct plant residue inputs, transforming those, but also how are they incorporating and transforming material that's been transported from another part of the landscape. And so what is the balance of microbial biomass to these different forms of carbon in the soil? And then how much carbon dioxide is being released upon microbial decomposition with respiration? And so I think perhaps a good way to look at that in an integrated fashion may be to look at parameters such as carbon use efficiency. So I draw that little purple box around what carbon use efficiency encapsulates, at least in this case. And so this definition of carbon use efficiency, if you're not familiar with it, can be widely variable. There's lots of different definitions and ways to measure carbon use efficiency that tell you different things about the temporal and spatial scale of microbial dynamics in a landscape. And so I don't know that we've necessarily settled on a specific definition for carbon use efficiency here, but this is what we're talking about. So I'm trying to think about how carbon cycle processes might vary across a hill slope. So looking at how both particulate and soluble material move from upslope to downslope. And there's lots of work that's already been done with other folks at IML-CZO on some of these parameters. I'm happy to talk about that more later. I don't want to get into too much detail because I'm going to try to wrap up here real quick. I don't have a very good sense of how much time I take, so I'm going to try to keep it short. And then also this vertical movement through the MIRZ. So this is a conceptual diagram that Ashley Deere put together and has let us all use, and thank you very much for that. But it shows how you can have these processes, different processes occurring with depth across the rooting zone. And sort of the major ones for CINet are plant rhizodeposition and maybe litter deposition as well on the surface that influence the amount of organic matter input into the soil. But you also have these tile drains at depth that are changing the redox gradient in the soil. And so fundamentally altering the types of transformations that are going to occur in these systems, where before they may have been saturated soil systems for much of the year, they're going to be unsaturated for now a much longer period of time. So that not only increases plant growth, but you've also got this introduction of oxygen input at depth that may be creating two different oxygen diffusion fronts in the soil. And that's been discussed in detail by the CINet group, but I think that's somewhere we're very interested in going. And so I see lots of potential collaboration with other folks and other projects. I haven't really talked too much about the measurements that we're going to make, but I already know from chatting a little bit with Emma that there are lots of complementary measurements we're already planning to make. So I think that's great. I think there are other systems where I would like to look at some of these dynamics, specifically these dynamics related to climate at the surface. That's really something that I've been working on off and on since I was a graduate student and postdoc. So these are really intriguing questions for me, at least. And I think I would like to be able to expand this type of work to these other critical zone sites. So I think I'll leave it at that. Does anybody have any questions or discussion?
Yeah, thank you so much. Anyone have any questions? Please remember to unmute yourself.
Yeah, Sean, this is Bruce. Good talk. Gave me a better sense of sort of where you fit, since you're new. I'm getting a sense, but it's great. We're all kind of learning about what we do. I guess one thing that struck me, I've been engaged a little bit in your input, and I think most of it is, as far as I'm aware, is really focused on kind of these uplands so far. And I'm a river corridor guy, sort of. I mean, I am interested in the uplands too, particularly with, like we've talked about, with this connectivity between the uplands and the rivers. But I don't know virtually anything about microbial processes, so this would be a great learning experience for me to learn more about this. But I know that when I talked to Aaron Packman a little bit, and I know he does some microbial work at Northwestern, one of the things that we discussed was if you get into the river corridors, how do the microbial communities change with the type of depositional environments you're getting? Because you have a lot of different kinds of depositional environments in the river corridor. I mean, an amazing array of them, actually. And some of them are, very young and very new. So, if there's a flood and that flood deposits material on a bar, and it's like fresh sediment, but I'm sure it's definitely got carbon in it, and it almost certainly has microbial action going on in it. But how does that differ from something that's beginning to be incorporated into more permanently the bar, and then into the floodplain, and might be very, very old, might be decades old, where soil's beginning to develop? And so maybe looking at these time transgressive gradients of deposition and how the microbial communities vary at these interfaces of not just space, but also time, I guess, as a factor. Do you know if anybody's really looked at that? I mean, you're aware of that kind of work, but I think it'd be intriguing to look at how carbon, microbial, and microbial activity are linked to these physical processes of storage in river corridors.
Yeah, there hasn't been a lot of work trying to quantify microbial dynamics across a landscape, but the work that has been done is mostly dealt with erosional deposition. Mm-hmm. So I'm not again, you're right. I mostly work in uplands, so that's the work I'm more familiar with. And sort of based on some of that previous work, the naive hypothesis that I originally came up with, sort of a general hypothesis, is that at any given point on the landscape, you've got perhaps maybe two types of input. You've got input that's coming from plants or autotrophs that are extant in that system, right? So plants may be growing on the floodplain, autotrophic bacteria and whatnot, inputting carbon. But then you also have all of that material that was transported there from somewhere else that represents carbon that is potentially older in terms of when it was input into a soil. And so sort of the naive hypothesis would be, well, that carbon, because it's been around longer, it's being continually transformed as it's being transported across the landscape. So you might expect it to have a greater proportion of microbially derived carbon substrates. And in another broad sweeping generality, we think of that microbial residue as being a little more complex than just undecomposed plant litter. And so that right there changes maybe the ability of the microbes in a floodplain, say, or a river corridor to transform that material. It may take a little more energy. You may find more specialized groups that are anytime things get more complex, you tend to see a greater degree of specialization. And I'm sure Emma can speak to this much better than I can. But that's what I came up with originally, based on the state of our knowledge. But there was also some evidence from IML CZO work that shows if you get a big enough event, say a precipitation event, you can rapidly move material from upslope all the way down into a river corridor, and it can be essentially the same age as that fresh input.
Right.
So that kind of upends the thinking, and it means we're going to have to, like you say, really look at the temporal dynamics and try to isolate them from the spatial dynamics if we can.
What about the
difference?
That's
not easy.
What about the difference in vegetation types? I mean, on the uplands, you have mainly crops. You have, predominantly corn and soybean, at least in the Sangamon. And then in the river corridors, they're all pretty much forested once you get down into the lower parts of the corridors. So the local extant input is from mainly trees and understory growing beneath those trees. And then, but the carbon up on the landscape is mainly in the form of these cultivated crops. Do you think that has any influence on the microbial communities, this sort of mixture of material that?
Yeah, it should, because when you've got trees with a lot of above ground biomass, you've got not just leaves, which we think of as relatively easily decomposable. You have all that stem, and tree trunk litter, lots of lignin and cellulose that are more difficult and take longer to decompose. And then you have a different amount of root input. And it varies a lot by plant species on how decomposable each of those types of litter is. I'm not so familiar with the tree species in Illinois, but when we've worked on corn litter decomposition, it turns out that, leaves are the most easily decomposable part of the corn, it turns out. Followed a little closely by stems, because the stems really aren't that much different from the leaves for corn since it's a grass. But roots have a different, they break down differently in the soil. At least in some of the systems that I've worked in, they're a little less easily decomposable compared to leaves and stems.
Mm-hmm. Let us move to Dr. M Aaronson now, and we can come back to continue questions, if that's okay, so that we have time for everyone. So, Dr. Aronson, you can go ahead. Hi.
Let me stop sharing.
Perfect. Okay, and I'll share mine. Give me 1 second. Oh, you know what? Let me try that again. Okay. Now, can you guys see it full screen now?
Yes.
Okay, perfect. And so I just wanted to thank you all for your invitation to be here and speak with you today. You know, I think one of the positive things that's come out of this terrible situation we all find ourselves in is that since we're working remotely from home, we can be working anywhere and we can be presenting with any group of people, and it's a pretty wonderful way to exploit the current situation. So I'm very happy to be able to speak with you today, and I hope that some of what I have to say will kind of build on and connect with what Sean was just discussing. It was really interesting to see your presentation, Sean. So similar to how Sean built his talk, I am going to describe some of the impetus for the new Geomicrobiology CZCN project, and kind of give you some of the context that we're working in, and then more verbally than with slides, talk a little bit about where we hope to go. So walking you through this slide, which is the only one with text, so I led a few years ago a big project across all of the CZOs where we worked with collaborators in each CZO to collect samples down to 1 meter or refusal, and send those back in depth increments of 10 centimeters. And then my lab analyzed those along with a bunch of other labs that did a suite of analyses. And we found some interesting trends. Now, heading into this exercise, we very much believed that we would see differences in microbial communities between sites, but that pretty much all the sites would show a trend of decreasing diversity and shifts in composition with depth. So the context going in was that we had a really clear hypothesis, and we wanted to know where those shifts were occurring. We wanted to try to understand how the different factors in each site might influence where the shifts in composition and diversity were going to be observed. For example, we've talked a little bit about the root zone as like a critical interface. We were curious, you know, will things drop off just as you get away from the surface, or does it have to do with how deep the root zone is? What are the different factors that the bedrock might influence in terms of how much of a shift there is going from the surface? Now, we just did one snapshot in time, and so we knew we couldn't answer all of those questions, but we were starting that process, I guess is the best way to look at it. We were very surprised to find that sites did two very different things. So some sites showed the gradients we were expecting. Other sites did not show gradients. We called them uniform sites because things were very similar. You go deep into the soil and things look almost identical to the surface. And so we really became curious about the different types of factors within a site, both above ground properties like vegetation and climate, as well as below ground properties like, you know, bedrock type, nutrient content of the bedrock, things like that, that might be able to influence why we see such differences. So let me kind of take you through, there's a chart here, a figure on the bottom right, which it is a principal coordinates analysis. So some of you might have seen a principal components analysis. This is not quite the same thing. What we're trying to plot here is all the different microbial communities that we see in all of our different sites. So I've categorized the sites as shallow or deep, and I'll show you in the next slide, or in 2 slides, how we came to those conclusions. But what's really interesting, I'm sorry, that's uniform or gradient, I apologize. Shallow or deep is just, you know, shallow is the first 30 centimeters and deep is the next 70 centimeters. Sometimes it was less than a full meter because they hit refusal before that. So about the next 70 centimeters, I apologize, I misspoke. So for the shallow soils, that's all blue circles, and all the deep soils are red circles. We see this big divide in that the gradient soils that are deep seem to differ from just about all the other samples. So these deep gradient soils, that's what we expected to see at all the sites, but there's many uniform sites where the deeper soils still look in terms of composition like the shallow soils from those sites and the shallow soils from all sites. There's a relatively high amount of similarity across shallow soils in many different sites when you compare them with the deep soils. The deep soils are so different, they make the shallow soils look the same, even though we know there's a lot of different soil types here, vegetation types, there's a lot of different microbial groups that are represented by each of these dots. So in reading this, you look at these different, each circle is an entire microbial community. And if 2 circles are close together or overlapping, they're almost the same in terms of composition. Whereas if they are separated, physically separated in the graph, then that means they're very different in composition. So just sort of giving you some context for how to understand what we're looking at there. Okay, so we saw these 2 really different types of sites, and in order to come up with what would be interesting to explore further at this real interface of geology, you know, looking deeper and deeper into soils, geoscience in general versus microbial community analysis and understanding of microbiomes in the environment, we really wanted to zero in on what would be driving these differences in variation with depth. So we also wanted to, in some ways, since we had been sampling at all 10 of the former CZOs, we wanted to preserve some of that diversity in location, diversity in the the geological history of sites, diversity in the types of soils that we'd be analyzing, but also we wanted to contrast shallow versus deep, gradient versus uniform types of sites. So we selected 5 of the former CZOs to set up in each of these locations 2 different types of sites within each location based on the previous data. So this is the data that I was referring to. So here we're looking at alpha diversity metric. And previously when we were looking at the figure, it was beta diversity. We're looking at how the different composition is between samples. Now it's alpha diversity, but we're looking at it with depth. Now, depth is going from left to right here. I know it's a little bit jarring to see it that way, but just follow me. Whereas diversity is on the y-axis for each of these, and greater diversity is higher, going down would be lower diversity. So some of our gradient sites are the ones generally on the right, where you can see that diversity, while it does bounce around in certain sites, there's, it goes high and low and high again. In general, the trend is pretty clear that in most of these sites, diversity, alpha diversity is decreasing with depth. And just for a reminder, that's what we expected to see everywhere, that we would see, very few overlapping organisms at the surface of the soil and deep in the soil, because they're doing very different things. There's more chemolithotrophy deep in the soil. There's some phototrophy at the top of the soil. So that's photosynthesis, but being performed by different microbial groups. You'd expect to see that only really at the surface. And there's also these trends where certain poorly described phyla that people see at depth, you'd expect them to really just be at depth. We ended up being able to do shotgun sequencing in this project and describe a new partial genome for a previously undescribed phylum that we really found mostly at depth in some of these gradient sites. And it was found in multiple sites, which is very interesting. It's got to be a very important microbe at depth that we don't really see at the surface. So these gradient sites are representing what we expect to see. The uniform sites on the left, shown in red, those you can tell there's very little change with depth. As you go from left to right, that's getting deeper into the soil, it's almost the same amount of alpha diversity. And surprisingly, in a lot of cases, that also corresponds to very similar composition. So, you know, things like the root interacting microorganisms, right? You see them in these sites just as much at the surface as you do further down in the soil profile. Things like photosynthesizers, even. We were very surprised to see cyanobacteria at depth in some of these uniform sites. We don't know what they're doing. We know they have the ability to fix nitrogen in some circumstances, so we think that might be what they're doing at depth, but so far we haven't been able to really isolate those functions. So we selected these 5 of the former CZOs because we feel that they'll help us to contrast and understand what's driving these trends. The types of measurements that we plan to do, obviously we're going to do a lot more sequencing. That's the type of data that I'm demonstrating now. And we're going to use not just amplicon sequencing, which is something that you know, is very commonly done, where you can look at one gene across many different organisms and try to understand their phylogeny and their relatedness. We're also going to do a lot of shotgun sequencing, and we've been in discussions. This hadn't been our plan, but we've been in discussions with some of the other CZCNs, or so some of the other clusters, and we've found that some of them are planning to use a new sequencing platform. It's called PacBio. What it does is it lets you get a really long set of genes, a really long DNA sequence from many different organisms within the same sample. And it'll allow us to better identify which photosynthesizers in particular, which cyanobacteria are the ones that we're seeing at depth. It'll help us to better understand really what are the exact microbes that we're seeing at different depths. And so we're excited about trying that out. And it's a great area for collaboration with some of the other clusters. We're also going to be we're going to set up sensor networks so that we can observe CO2 and oxygen concentrations at various depths. Right now, we're in the process of figuring out exactly which depths, which is a controversial decision within the cluster. But we're also going to look at, you know, more basic parameters like soil moisture and temperature and how those vary with depth. And then over time, we're going to take many samples at the same site at different depths and be able to try to contrast things like enzymes, which is mentioned by Sean as well. You know, I think it's really important to understand enzyme properties and enzyme, you know, shifts with depth and how they correspond with what we observe in terms of the microbial communities and the functions of those microbial communities. Connecting microbial community structure to its function is kind of the next frontier in microbial ecology. I think everyone to some degree is working on that. And by looking deep in these soils, I think we have a unique view on those. So a lot of the measurements I think will be actually pretty similar with CINet. I hadn't known how to say that out loud. I think that's what you said, CINet. But I think, you know, we're not looking at actively managed landscapes. We're going to be looking at, for the most part, these are undisturbed landscapes. In some cases, like in the Calhoun CZO, they're areas that had previously been extremely disturbed, but have been allowed to reforest and go back to a natural state over time. We're very interested also how that process might have influenced the structure of the soil community with depth when it had been cropland and no longer is. And I think that's something we can really work with CINet on as well. And so just to sum up my talk, I don't know how long I've been talking, so stop me if I've gone too long. But this is kind of how we're setting up these specific hypotheses that we're addressing with our approach. With the gradient and the uniform sites, we're addressing essentially the same hypotheses. I'm sorry, we're addressing the same overall goals, but our hypothesis is that the deeper processes, bedrock, lithology, soil type, hydrology, groundwater, that all of those factors are going to be less relevant to the soil microbial communities at the uniform sites, and that's why they're uniform. So that's the hypothesis that we're going to test by contrasting these different types of sites. We think that in the uniform sites, it's the plant dominant characteristics, what type of plants are there, what type of root exudates are they releasing, their interactions directly and indirectly with different microbial groups, as well as climate and weather. We think that those combination of things that we have on the top here as being surface-based processes are going to dominate the structure and function of the microbial community, and that biogeochemistry of the critical zone in these ecosystems. Whereas in the gradient sites, we actually think that bedrock, lithology, soil type, hydrology, and groundwater, all these deeper surface processes are going to exert a very large influence on the microbial communities and their functions. And so how that plays out in each site, though, I think is going to be very different. We've listed a number of factors for the surface and for the deep influences, and we don't know which ones or in what combination they're going to be important in determining the microbial communities at depth. What we do know is that, we've really done a lot of literature review. We've all done a lot of research ourselves. We're pretty sure that people don't have a grasp on this. It's not that we just don't know, it's that nobody really knows what types of factors and in what combinations can you really see sites that do not change the microbial composition with depth. We'll also, and this might be something you guys have thought of as I'm talking, we'll also just, by going deeper than 1 meter, I think we might be able to get at some of those answers right off the bat. So we're excited about that opportunity. We, had limited funds to do what we did initially, and we're relying on collaborators to take samples. And it's a lot of work to dig a pit that's deeper than 1 meter. I think if anyone here has ever dug a soil pit, it's a lot of work to get to 1 meter. And so, we're going to go as far as we can. We're probably not going to bring in equipment to go, as deep as possible in these sites. We'll probably still do a lot of hand digging unless we can coordinate with others to dig deeper with equipment. But we are really excited about the opportunity to look at that very deep soil, especially in some of these uniform sites, and see, do they become gradient sites if you look deep enough? And that's another part of this hypothesis.
Thank you very much. Very nice presentations, very interesting work. And to open the room for discussion now, I would actually be personally very curious to see how this diversity of microbial activity and identity changes in IML CZOs, where we have installation of tile drainage. We have a significant coverage of those tiles, which add oxygen to the system at different depths, and they remove water. So I would expect, I don't know if Dr. Schaeffer has any feedback on that, or anyone?
Oh, it's something I'm very interested in as well.
Yeah, we initially we did have 2 IML sites, soil pits that we collected samples from with collaborators at IML. And so, I could take a look at those. Actually, that might help us to explain if those are uniform sites, that might be a good reason why we're observing that. If they're gradient sites, if they're not uniform sites, now I would be actually surprised given that information, because I didn't know that.
Yeah, and the alpha diversity may even go the other way with depth, may even increase, probably. Who knows?
Right, because if you see, the chemolithotrophs at depth, which are processing, some of the complex carbon, they're processing some of the minerals that are, partially released from the bedrock and things like that at depth. And then you also have aerobic microscopic organisms that are performing a whole suite of other transformations, that just being at that interface and allowing for that, yeah, you might actually see greater alpha diversity with depth. I'd be very curious.
Mm-hmm. Any other questions? Yes.
Yeah, I have a question. So I'm a geomorphologist, so I'm very new to all of this. So please excuse my question if it's really ignorant. But how does the sort of geomorphic setting of these sites vary? Is there anything systematic about the different patterns with, you know, are you in erosional or depositional places or?
So that's a good question. And right now, so the locations we've identified, so I can answer better about our planning for the future than I can about the data that I presented just now. So for our planning for the future, we've got these 5 locations. And within these locations, we're working out right now where our actual sites are going to be. One possibility would be to try to contrast a more erosional with a more depositional location within, let's say, most of the sites. It may not be possible at every location, but that's one thing that we are considering. Several of these are on hill slope environments, but not all of them. And we're thinking about how to use that to our advantage in terms of contrasting and comparing between. Yeah, but I think there wasn't, so that the other answer that's about the data I already have presented is that I believe that we looked at those factors as a possible explanation for the uniform versus gradient. I don't believe it emerged as a likely dominant driver, but it probably contributes within a location to the site-by-site differences in terms of these things.
And then I guess another really naive question, but when you find these different organisms at different depths, does it have to mean that they're living there, or can they just kind of be there?
That's a great question. So the way that we characterize microbial communities, my lab is almost all culture independent. So that means we don't try to grow things in order to understand them. And that's a conscious decision because so far, people can only grow in labs probably, they say 1 to 5% now. I don't know exactly what the current percentage is, but it's a really low amount of the total diversity that's out there. So when you focus on things that you can grow, you lose that perspective of the whole diversity of things that don't grow. But there isn't a great other way to establish what's alive versus what's just present there. And so what we do is DNA sequencing, where we try to, just characterize the community from the DNA that we see. But depending on various factors, DNA can last for a long time. We don't specifically focus on relic DNA. And there's, usually methods people try to use to get at the DNA that was left behind from organisms in the past. We're not doing that, but we're probably getting some percentage of relic DNA showing up incidentally. It's just what, is happening. And so I can't say for sure that everything that we see is currently alive. I can say that in many cases, it's organisms that theoretically could live in those locations, even with the gradient and the uniform. We don't know what the cyanobacteria were doing with depth. That's kind of the big example of something where we're having a little bit of trouble explaining it, but it's so abundant at some of these lower depths that it can't, or I assume it's not just a relic. I think it's real, but I don't know exactly what's explaining it. If it was just, really rare, I could say it's probably just relic DNA. Yeah.
Thanks.
Does that approach your question?
Oh, for sure. It's this. I guess my impression of this is that it's kind of like there's so much still to understand here. I mean, just the fact that you're like, we found a whole nother set of things by doing this is like, yeah. I guess it's fascinating to me. I think there's so much out there to still learn. I guess more broadly, maybe this is a question kind of for Sean as well. I'm really interested in how well, like where the field is in terms of connecting between the development of soils and the microbiology part. And so, you know, we have these snapshots of which microbes are in what places right now and how that relates to what the soil is we have now. But I guess I'm really interested in sort of all this has to integrate over time to kind of give us the soil, or I don't know, I guess a lot of the parts of the soil come from the biology over time. And so I don't know. I guess just kind of thinking about, I'm really interested. I'm working with Bruce in the river corridor thing too, but one of the things I'm interested in is sort of how do you go from this newly deposited stuff to it becoming a soil, and over what time scale, and that presumably you have microbial communities that are going to have to facilitate that change and maybe change themselves over that time scale. So I don't know, is that just completely wacko as a thing people are working on? Or
I mean, I think that you're talking specifically about, you know, the deposition of new material and how that becomes part of a soil profile. And I think depending on how you look at it, the new material, it's soil right away, right? I mean, it's material that's some combination of mineral and organic components. There's already microorganisms that are living and probably thriving in tiny little aggregates, you know, microaggregates that are part of that material that was deposited. And so I guess for me, it's not thinking about it, the microbes being part of the transformation to make it soil, as much as once it's deposited, what happens next, right? So there's some sorting in the soil profile, you know, microbes that you know, they might have been in this newly deposited material, but they're happier further down. They might kind of let themselves drift. There's other microbes that might try to live at the surface of the soil. There's some stratification. And then along the way, if that is undisturbed for long enough, then you get a more defined soil profile that will develop over time, where, you know, you won't have the fresh material won't look so different. Let's just say, you know, you'll see the of whatever the layers, the strata of whatever type of soil that is. So it's interesting to think about that interplay. I think a lot about other types of depositional activities. So I think about microbes that are deposited by wind a lot, and we did some studies in the Sierra CZO, looking at the dust coming into the Sierra, and we found that some of it was coming from really far away. But some of my colleagues were really focused on the nutrient component. And so that dust is bringing in the Sierra Nevada and California, probably most of the phosphorus that's used on an annual basis, which I found really fascinating. And so once that dust arrives, there's microbes getting in there, you know, they're releasing compounds that can grab that phosphorus right away. And so I'm sure the same thing is happening when you get deposition of material, you know, from river environments and all sorts of different types of depositional areas. So I think you're bringing up a really interesting point and sort of set of questions for research.
Yeah, I would just add that there are a couple people out there actually looking at how organisms in the soil drive chemical weathering rates in the soil. Okay. So you can actually, well, at least they would say you can actually see biological activity, the rate of biological activity directly driving chemical weathering on an annual scale. And that's something that people really haven't been thinking about in terms of soil formation.
That's super cool.
I mean, yeah.
Yeah, we should talk
and
about
I don't know, I don't know much more about it than that. So I can't answer any more questions than that.
Anyway, thank you both for some very nice presentations. I learned a lot.
Are there any other questions?
I have a quick question for Emma.
Sure.
you saw these differences in alpha diversity between gradient and uniform sites. Did you have an estimate of just overall abundance of organisms, and was it different between gradients and uniform sites with depth?
So abundance is a tricky one. I'm going to say I don't have data that I'm sure of that can help me contrast these things. So that my biggest answer is that I don't have reliable data. There's a lot of disagreement over what type of measurement to use for abundance. Anyway, so just leaving it there, I'm not 100% sure. The DNA sequencing, which is very much a biased experience or process, it did seem to show lower abundance with depth overall, in particular in the gradient sites. In the uniform sites, I think it still showed some lower abundance with depth, but it's very difficult to extract DNA from deeper soils. It's just more difficult than surface soils. And part of that is, we think, because of lower abundance, that there's just fewer cells, you have to work harder to get them out. But it's so then with the sequencing, we may be overrepresenting the microbes from depth because we're putting in more effort, let's say, to extract them, and then we try to have the same amount of DNA from each depth increment in our sequencing runs so that we're not biased too much towards the top. I think we end up biasing towards the bottom. So I'm going to say probably abundance was lower in just about all the sites with depth. Yeah. That's something we're going to try to make sure we have good standardization of methods for this project we're embarking on now, so that we can answer that question more directly, because I would have liked to have a clear answer for you. Yeah, it's a good question. It's one people are very interested in.
So if there are no other questions, we will thank you again. It has been very nice discussion, and we'll see you all next month in the next pop-up webinar. All right.
Thank you, Emma.
Thank you.
Thank you.
Thank you.
Thank you.